Okay, is not the splash screen, by the way.
Hello, everyone, and welcome to today's Keynote webinar titled The Path to Universal Newborn Sequencing. This webinar is a part of the 11th annual event in the Genetics Week virtual event series. I am Kaylee Bach of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. And with that, I'd like to now present today's speaker, Dr. Robert C. Green, Professor of Medicine Genetics at Harvard Medical School, Director, Genomes2People Research Program, Preventive Genomics Clinic, Population Precision Health at Ariadne Labs, Mass General Brigham, and Broad Institute. For a complete biography on our speaker, please click on the presenter window that's located directly below the media player. Dr. Green, you may now begin your presentation.
Thank you so much. I am so excited to be talking today about the path to universal newborn sequencing. And I'm particularly excited to talk about this because I think it's going to be relevant to anybody who has a baby or who's going to have a baby, or maybe who has had a baby or been a baby in the past. In other words, everyone, because I think this is the real tip of the spear when we're talking about bringing genomics into day-to-day practice of medicine. Let me see if I can set up my slide here. I am, hold on, flip that button to me one more time. There we go. All right, great. And I'm still seeing the slide 23 in my large box. But if you're seeing slide number 1 and my next button is not working. Any suggestions on that? Okay. All right. Let's try again. Okay, there we go. All right, thanks very much. Sorry about that delay. So I'm Robert Green. I'm a medical geneticist and implementation scientist who's been working on the question of how we integrate genomics into the practice of medicine for the last 15 years or so. And I'm going to talk to you today about our latest research in newborn sequencing. I would like to say that I'm supported by NIH grants and a number of other funds. I do consulting work with the entities that you can see there, and I've co-founded 2 companies, as you can see there. Now, the work I do is really facilitated by a core group of people that we call Genomes2People Research Program. And these are a group of geneticists, genetic counselors, implementation scientists, economists, and psychologists who work on the question of implementation. But for the MedSeq and BabySeq projects I'm going to talk to you about, we have had a large number of collaborators and really too numerous to list. So I'm very privileged to be presenting today on their behalf. For those of you who are listening and who are trainees or have connections with trainees who might be interested in this area, I want to mention that we host trainees, particularly during the summer months. who either come to Boston or work with us remotely as trainees. And we do this at all levels, from mostly college age through junior faculty. So if something that I talk about today excites you, please feel free to get in touch about that. And finally, before I jump in, I think we all recognize that in genomics, there's a risk that many of the databases have been created with persons of non-African ancestries, non-Hispanic ancestries, persons primarily of European ancestries. And I'm very proud that our programs have been among the first to offer elective sequencing to persons of color, and most recently to offer whole genome sequencing to African American and Hispanic newborns. So as an entree to talking about newborn sequencing, let me just spend 1 minute on the project we did before that, which we called the MedSeq project. And this was the world's first randomized trial to offer comprehensive genome sequencing in healthy adults. Now, healthy adults, by definition, don't have what you would think of as a genetic syndrome, so you wouldn't necessarily expect to find any mutations in those individuals. So it was absolutely startling to us that we found that 20% of the number of people that we comprehensively sequenced and looked at over 5,000 disease-associated genes had a mutation or biallelic recessive mutation associated with disease. And this was completely startling. We've published quite a lot of papers on this, and we think that, in fact, some of these represent cases of genetic disease that were underdiagnosed or misdiagnosed, and some represent a risk factor for things that might still occur in the person's life. So we coined a new term for this, which we called monogenic disease risk. It's not a diagnosis because in most cases, the person hasn't actually developed the condition yet. But if you're carrying a BRCA mutation, you have a monogenic disease risk for breast and ovarian cancer. If you're carrying a Lynch syndrome mutation, you have a monogenic disease risk for colon cancer. And so this became a new way of thinking about disease. And you could extend that not just through the monogenic and biallelic disease risk on the far left of this slide, but also through recessive carrier states, where 90% of people are carrying at least one recessive carrier that could contribute to devastating disease in their children. About 80% of people are carrying an atypical response to one or more medications, and this newest form of polygenic analysis revealing polygenic risk scores where fully 50% of people may be carrying an elevated polygenic risk score for common complex diseases like type 2 diabetes, non-hereditary breast cancer, non-hereditary colon cancer, and so forth. So there's very much a wealth of information in the genome. In part, we haven't been using this for population screening because these different bins have been all separated from each other. Now, what happens if you imagine the concept of population screening? We've been talking about this for a long time, but what if everybody in this audience had their genome sequenced? Why would that be a bad thing or a good thing? Well, it's a good thing because you can create an awareness of possible unsuspected genetic diagnoses, as we found in some patients in MedSeq. You can risk stratify and find those people who are at risk for cancer, sudden cardiac death, and other genetic syndromes that we might want to surveil more carefully. And because more and more people seem to want to know what they're at risk for, whether or not they can take measures to mitigate that risk, or even choose to take measures to mitigate that risk, they seem to want to know. So why wouldn't we do this? Well, the main reason is that individually, these are rare conditions and the prior probability of each one is low. And so we're going to identify even if we do it perfectly, we're going to identify a lot of people who are at risk, but may not get the condition for years or may not ever get it because some of these mutations are not 100% penetrant. They don't actually cause the disease in everybody. They cause it in some. And because of these, it's very hard to accumulate clinical utility and cost effectiveness data. And then if you were to identify lots and lots of people with, oops, says okay. If you were to identify lots and lots of people with this, there seems to be inadequate expertise in the medical workforce. So this brings us to the BabySeq project, because if we felt, as we did, that this was valuable in apparently healthy adults, what could be the value in apparently healthy children who had even a longer period of time in which to express genetic disease, particularly the child onset genetic diseases? Now, this is particularly pertinent to our time because at this moment in history, we have a system that carefully and universally does screening, the heel stick test, on every baby born in every hospital in the United States, and for the most part around the world to some degree. But that system is overburdened, stressed, not easily able to add additional conditions. And at the same time, there has been an explosion in cell and gene therapies for novel genetic conditions. So that system we have in place cannot keep up. Moreover, it's a system that's basically unconsented. Parents, really, the baby's whisked away, the heel stick is done, the test is sent to the state lab, baby's brought back. Parents are really only involved if a possible positive finding is found. And no one thinks that we should enter into the era of newborn sequencing in this unconsented manner. So how can we start to explore consenting, communicating, choosing which genes, and actually implementing newborn sequencing? And that was the purpose of the BabySeq project, which we got funded by NIH in 2015. Now, again, the rationale for this and the urgency for this is that there are over 26,000 babies born per year with rare diseases that are not detected on conventional newborn screening, and probably over 100,000 that are born at risk. And once again, these numbers of treatable conditions are exploding in ways that far exceed the ability of the public system to keep up. So the BabySeq project was a randomized clinical trial in which we took volunteer families and randomized them to a sequencing arm and a control arm. And everyone had multiple visits, and the sequencing arm offered family history and a comprehensive genome report, whereas the family history arm only offered family history. And then we followed them for medical, economic, and psychosocial outcomes. Now, how did we pick the number of genes that we were actually going to look for? Because you can do a sequence, you can do an entire sequence, but the second step is you have to select a number of genes, 5 genes, 10 genes, 50 genes, 100 genes. You have to select a set of genes that you're going to analyze and report out. We ended up selecting about 1,000 genes, which were mostly more highly penetrant, younger age of onset, and genes with very good disease gene validity. So we're trying to minimize the possibility that we would identify a variant that was not going to develop into the disease in question. Though, of course, we understand that that will happen. This brings us to, I think, the question of how proactive or population sequencing is very different from indication-based sequencing. As I'm sure this audience is aware, right now, genomics is one of the most exciting ways to diagnose a mysterious illness. If you've got a child in the neonatal intensive care unit, or you've got a mysterious disease that you can't figure out, sequencing can actually give you a molecular etiology roughly 40 to 60% of the time. And that's remarkable and gives you tremendous insight into prognosis and sometimes into treatment. That's the use of genomics as a diagnostic aid. But we're talking about flipping that on its ear here and using genomics as a screening paradigm. And in that context, we want to make sure that the genes we are looking at have strong validity, that the variants we're looking at have strong pathogenicity, and we want to better push this toward actionability. The most important variants to identify first will be those that underlie treatable conditions. So I think it's valuable, again, to think about when someone is ill and you're doing a genome, you're doing it in order to determine a molecular cause, sometimes called a diagnosis. But when you are sequencing an apparently healthy baby or adult, what you're really doing is most of the time you're looking to determine a risk variant. And so I would call that risk stratification, not diagnosis. And in the same manner, I would stay away from language like false positives. If you find, just to take the obvious example, if you find a 25-year-old woman with a BRCA1 mutation, a classic BRCA1 mutation, you haven't diagnosed breast cancer. What you've done is you've recognized that this woman is at dramatically increased risk of breast and ovarian cancer, and you have now hopefully committed yourself to offering her enhanced surveillance, prophylactic surgery, and the like. Now, when you ask parents if they want newborn sequencing for their babies, they overwhelmingly say they do. When you go in with a 27-page consent form and you tell them they have to come back for a few research visits, it turns out to be a different story. But somewhere in between, there's a sweet spot where you can consent parents in a reasonable manner and then work through the friction that would allow this to occur without huge hoops for them to jump through. So that's an interesting lesson in and of itself. And what did we find when we did this? Well, we found that with about 1,000 genes, 11% of these apparently unaffected babies had a mutation that was associated with a treatable condition. And for some of these, there were features of the disease which were not properly recognized when we did the DNA testing, and so were recognized in retrospect. And for many of these, there was additional family history that came out as a result of the DNA first. And for some of these, there were no features of the disease apparent, but there was a sense that these children were now at increased risk. Now, you might say, well, did you find anything else out? Well, yes, in looking for biallelic recessive carrier traits, we found that these babies were, of course, carrying heterozygous traits. And they had many, many heterozygous traits, including more than you would get from any commercial panel. So that was interesting of itself and potentially useful to those families for having their next baby. But you also might ask, well, did we distress these families? Did we create anxiety, depression? Did we disrupt parent-child bonding? And that's where our randomized clinical trial format really shines, because we can show, and we've published this in JAMA, that we did not. In fact, if you look at the bars correlated there with the control group and the neonatal newborn next generation sequencing group, and even the subset in whom we found monogenic disease risk, you can see that on each of these different validated scales of depression, anxiety, and so forth, there were no differences in the average scores that these parents received. So this is an opportunity to say we have been over-indexing on distress and under-indexing on the possible medical benefits of sequencing. Now, you might also ask, well, are you going to cost the healthcare system a whole lot of money? And the truth is, we probably are going to cost it more, but perhaps not too much more. Here, we actually were able to track the costs in the 10 months following the sequencing between the two arms of the study, the group that was sequenced and the group that was not sequenced. And you can see that the group that was sequenced, indeed, somewhat greater spending was made than the group that was not sequenced. Moreover, if you look at the subset of families in whom there was a monogenic disease risk found, you can see that once again, there was additional costs, but it was not a huge amount of costs. And then the most startling thing about this was that everything we found was treatable. We compared all of these items on a semi-quantitative scale that ClinGen uses for actionability. And all of these conditions had concrete actions that could be taken to either treat or surveil for early detection of the disease in question and for better outcomes if you detected that earlier. Now, what we didn't expect, and what you can see in this slide, is that we not only stimulated surveillance and follow-up in the babies themselves, but there's something magical about the fact that these were discovered in babies. And this information was distributed and percolated out through other members of the family. And in other members of the family, we ended up generating specialty consultations, imaging studies, ongoing surveillance, additional genetic testing, and most startling of all, as you're going to hear from one of the parents themselves, several of these babies were discovered to be carrying adult onset cancer predisposition genes, which normally you wouldn't be returning in babies. But because we did return them, we were able to track these up into the parents. It so happened by coincidence, it was 3 mothers. And each of those mothers had risk-reducing surgery in the 3 to 5 years following this disclosure. I mean, let's pause for a second and think about that. The current standards of ethical genetic testing say that you should not give adult onset risk variants to families about their children. In other words, we had no business giving those families the information that their baby was carrying a BRCA1 mutation or a Lynch syndrome mutation. But when we did that, because we were in a research protocol and we could do that with proper IRB permission, when we did that, we inadvertently managed to give the rest of the family this information, including the fact that the mothers were carrying these mutations, and they had no idea that they were carrying these mutations otherwise. So when they found this out, they were able to take action, and in all 3 cases, we believe that prophylactic surgery may have certainly saved one or more of their lives. So in sequencing these babies, we were arguably able to dramatically improve their lives, not only directly through surveillance of their own mutations, but indirectly, but powerfully through the possibility of life-saving information for their mothers. Now, the BabySeq project, I don't know if anybody on this webinar has actually heard of it, but we've gotten quite a bit of notice because it's been a fairly daring enterprise. We've done something that people have been predicting should be done for years, but no one was quite sure how to go about it, the cost, the communication, the choices that had to be made. So I'm really proud that we were the first in the world to do this. And I'd like you to hear just a couple of minutes from the voices of some of the BabySeq parents and their impressions of the experience. Let's see if this works when I push play.
We just thought, like, why isn't this done for everybody?
I think part of the attraction of participating in the study was having that information and how empowering that information would be.
Let's be real. I mean, when you have a newborn, it's kind of like your whole world changes. So knowing a little bit of information is going to be helpful down the road.
We just want our children to be healthy and happy. And if we can know that what could potentially be harmful to them, we can hopefully act on it.
I got involved when my son was born in January 2016. He's now 6.5. When we got the results for my son, we had learned that he was a carrier of something called supravalvular aortic stenosis. I learned that it can cause a narrowing of one of the valves in the heart and potentially could cause significant health issues for him down the line. You know, you think about those high school football players that just fall over and die in a football field because they had a heart condition that nobody knew about. And then so I was like, oh, well, if they had only known, if there was some way to know. So I always kind of comforted myself by thinking, at least I know it.
The most critical thing that they found with my son is that he has the BRCA2 mutation. I was told that I had a significant mutation that is a high propensity for a couple different types of cancers and that I should start to look into that. I would rather my boys have me for longer than me get something that I know is a higher risk for me. I'm not willing to accept the risk of cancer and leave my kids motherless. And so overall, just so happy that we have opted in, that we know our risks, that we're taking action steps to, extend our life.
You already do some screening, obviously, like by default. Right. So this was just, to me, this was just like an extra feature of screening, of being able to see more and kind of dive in more and be able to know more, ultimately, so that you are prepared.
It's just, in the long run, it's more beneficial to our society, to our world, to our history.
You don't see a lot of people with color. You don't see our demographics being represented. We have to count, and that's why we did it.
It was a really positive experience for our family, and we would participate again if we had the opportunity.
It has been a journey that we are happy that we opted into.
And I just felt more so empowered to have the information than not. And I think that, with everything going on in the world, with all our resources, we should know more so that we can do our best for our kids.
Great. So you heard some of those voices, and these were voices from our very first, essentially, BabySeq 1 study. And we've now been funded by NIH to expand this to multiple sites in what we're calling BabySeq 2. And in this, we want to particularly make sure that we are enrolling a diverse cohort. And to do that, we have selected sites throughout the country where we can be assured of a more diverse population to be enrolled. And we have worked with groups in the community to create culturally appropriate materials, train pediatricians, interview parents, and partnering with community leaders so that at a minimum we'll enroll 1,000 diverse newborns, but perhaps as many as 5,000 to 10,000 as other groups join us. So I want to put this out there to anyone listening. If you are associated with a pediatric practice that is seeing newborns, either through a private practice, a healthcare center, an academic healthcare center, and you would like to be part of BabySeq, please let us know. At the moment, we don't have extra funding, so several sites have actually managed to come up with some of their own funding for this, but we may be able to rustle up more funding if there's more appetite for additional sites and more families. Obviously, the more families we enroll, the more data we collect, the better we're going to be able to ascertain the medical, behavioral, and economic outcomes. Now, where are we going with all this? Well, our thesis is that genomics is going to be a lifelong resource. Some people are talking about starting even during pregnancy with fetal DNA analysis, sort of an extension of NIPT. But for us, just starting at birth is a powerful opportunity to screen babies for what they may be at risk for during their young life, their childhood, adolescence, early adulthood. And as knowledge is constantly evolving, this can be updated and upgraded in order to tell you at appropriate ages what you're at risk for. But also because knowledge is constantly changing, it may be possible to hold this genome in a cloud, say, and on a regular basis reinterpret it. You wouldn't even have to go back to the person for a new sample. You could simply electronically interrogate that genome as the databases and the science underneath genomic disease continues to change. So that's a really exciting vision. I think you'll be hearing more and more about that vision as time goes by. And it is a vision that we hope will power not only genomics, but the whole practice of medicine, which currently, through the perverse incentives of our healthcare system, is really focused on procedures and throwing a lot of resources at people who are already very ill. If we can somehow flip that and offer more resources to predictive and preventive care, then we can keep people healthy, keep people from getting sick, in extend the lifespan, extend the health span, that period of the lifespan in which we are healthy and robust, and everyone can live healthier, longer lives. So that's, I believe, where we're going. Genomics is obviously not the only tool in that toolkit. There's all sorts of self-monitoring devices. There's new initiatives to regularly scan people for risk factors. There's a constant tension between the question of picking up incidental findings and over-diagnosing, over-treating, over-medicating people, as opposed to early detection and saving them, which we have to negotiate. That is the business of public health, is to negotiate those things. But I think that with all of the technology that's being developed in this arena, the wearables, the smartphones, the smart diapers, the microbiome, multi-omics, genomics itself, DNA analysis is actually one of the simplest and one of the earliest and sort of the tip of the spear as we move into hopefully an era of prediction and prevention. Now, when we were first talking about our results, or even just doing the project at national meetings, we got a tremendous amount of pushback from the community. This was seen as radical, unwarranted, and perhaps in some of your minds it still is. I hope that the data we're evolving is actually reassuring and hopeful in terms of the possibility of integrating this. But gradually, other sites around the world have joined in this and are starting to consider doing newborn sequencing. And to support all of us, I co-founded something that we now call the International Consortium on Newborn Sequencing, which is a multiple group of investigators, starting with about 8 investigators, all of whom were starting this year to do or to plan for newborn sequencing at their own institution, including, of course, the country of England that's going to commit itself to a grand experiment of sequencing 100,000 newborns. And we had our first meeting of this in Boston at the Science Museum. Oh, there we go. And in that meeting, we had over 400 people attending, incredible speakers, including the CEO of Genomics England. And we really launched a sort of fellowship of scientific interchange and sharing between the key groups around the world that are attempting to begin work in this area. And since then, of course, another dozen countries and investigative teams have started exploring this space. So we're very proud of this. We've created a steering committee of the first 8 groups. We're having a monthly meeting, which everyone is welcome to. Anyone can join this who's interested, either as an individual or as an institution. And if you're interested, please go to that website, www.iconseek.org, and communicate with the executive director, Nick Encina. And I will say that if you're really interested in this, put it on your calendar now for the next annual meeting of the ICONS conference in October 5th and 6th in London at the famous Royal Institute, where such notables as Galileo have spoken in the past. Now, I'm going to wrap up shortly, and I can't wait to hear some of the questions that you might have. This is always a fertile area for discussion, so please don't hold back. But I also want to make you aware of one more paper that just came out a week ago. And this is a paper that was led by Nina Gold, my colleague at Mass General Hospital, and involved actually surveying over 250 rare disease experts about whether they think the time is right for newborn sequencing. And spoiler alert, nearly 88% of them do. And which exact genes should be the very first ones that we go for. And they were able to, we were able to explore 651 genes with them. And we created a histogram of the genes that had the greatest consensus. And over 400 genes were endorsed by over 50% of these rare disease experts. Really extraordinary amount of concordance in those. And if you're interested in what the top genes were, the 8 genes with greater than 90% expert concordance were the ones you can see here, with the number one being OTC, ornithine transcarbamylase deficiency, which is a disease that can be fatal at almost any age, if not detected, when someone eats too great a load of protein and becomes hyperammonemic. And all you have to do to save a child's life or to save an adult's life is know that they are carrying a mutation for this and adjust their diet accordingly. So it's a devastating potential disease with a very simple way to treat. And it is not currently looked for on conventional newborn screening. And it's not that rare either. So with that, I'd like to wrap up and tell you that I think we are heading into an era of genomic medicine. The cost continues to go down. The amount of information that you can extract out of a genome continues to go up. We have to adjust our thinking so that we're not thinking about diagnosis, but we're thinking about risk stratification. And I'd like to leave you with a couple of resources here, acknowledging the extraordinary team I get to work with every day. And you can read more about us and our work at the website genomes2people.org. Excuse me. Please follow us on Twitter, as you see there, or Instagram. And if you have any questions or would like to follow up for being a BabySeq 2 site or some sort of trainee experience, please feel free to email me. So with that, I hope we can open it up for questions.
Yes, we absolutely can. Thank you so much, Dr. Robert Green, for your informative presentation. We will now start the live Q&A portion of the webinar. I want to remind our audience that if you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. All right, so let's get started. Dr. Green, we've got some great questions coming in from our audience, and this first question has a few different parts to it. It asks, do you have any concerns with this information falling into the hands of insurance companies? What ramifications do you see coming to light if this were to occur? Not all conditions discovered are actually either treatable or actionable. Have you examined the nucleic acid methylation patterns of the newborns compared to the parents?
Great. Okay, super great questions. So let's just take them one by one. Everyone everywhere is concerned about privacy and discrimination with genomics. And particularly in the United States, there is a law protecting any discrimination from employers or health insurance companies. And that law is very straightforward and highly respected. But that law does not cover long-term care or life insurance. So theoretically, insurance companies could use this. And then the laws are very different in other countries. Actually, probably the most coherent in the UK. But it almost doesn't matter what the laws are. People perceive correctly their genetic information to be private, and they want it kept private. And so, yes, there are concerns about privacy and discrimination. I guess my position on this is that we have nonetheless over-indexed on those concerns and under-indexed on the medical benefit. I don't qualitatively see a difference between the privacy discrimination practices that could accrue for mental health information, HIV status, or other kinds of medical information and genomics. And this is, of course, part of the exceptionalization of genomics, which I think may have done more harm than good over the past 25 years or so. Now, if insurance companies did, your question about the ramifications, if insurance companies did use this, and a few have attempted to ask these questions in the US, but very few so far, I think it risks a backlash, and the insurance companies know this. So I think they've been very ginger in their questioning about genetic testing. Your statement about all conditions being neither treatable nor actionable is, of course, true if you're looking across all genomic conditions. And I guess when I talk about whole genome sequencing or whole exome sequencing, that possibility arises. But what I meant to say was that in each of these situations or scenarios, certainly in BabySeq, we are not returning all information. We're returning only genes that we think have a clear-cut actionable condition. And so I think it comes down to the genes that you select and your definition of actionable. And then finally, you ask about the methylation patterns. I think it's a great research project. And in fact, we've been talking to some epigenetic specialists, but no, at the moment, we haven't done anything with that. If you think you've got some funding for us on that score from Sanofi or elsewhere, we'd love to talk about that.
Great, thank you. Another question I have for you here. Do you think newborn sequencing will become approved generally? And do you think ethical rules will move towards a wide approval in other areas of the world?
I do think that this is going to, there's going to be a lot of pressure to allow people to pay out of pocket initially for this, which of course is the way a lot of technology gets spread initially, but also may not feel fair or in comparison to the current universal newborn screening. I think some areas of the world are going to be more aggressive about this. The Middle East is talking very aggressively about newborn sequencing. The UK is moving quickly toward this. Other traditionally European countries are a little more conservative, although the EU has, I understand, designated 42 million euros for a pilot study. So I think this is happening. Yeah, I think this is happening throughout the world.
Great. Next question here asks, what are the possible negative effects or limitations of using imaging studies on newborn genomic sequencing?
I'm not quite sure what's being asked here, but I take from it two things. You could find a child who's at risk, let's say for childhood cancer, and then you could do some additional imaging, let's say for risk of renal cancer, and you could find a ditzel on there and subject that child to what would turn out to be an unnecessary biopsy or surgery. So you can imagine all sorts of downstream sequelae that would expose children to more radiation if it's recurrent X-rays, or just to the possibility of finding incidental findings that would result in follow-up testing, and some of that follow-up testing could be discomforting or even dangerous. So I think that's I think that's an issue. But perhaps the questioner is also asking about the parallels between whole genome sequencing and whole body imaging. It's a parallel that's frequently alluded to. And there's merit on both sides in both cases. If you do a whole body image, you will often discover something that then requires to be followed up, and in that follow-up, you can spend money from the medical commons, and you can actually potentially put people at risk for things that turned out to be utterly harmless. Same thing is potentially true for a whole lot of findings on the newborn genome. And I think we're going to have to carefully feel our way with thoughtful follow-up to not over elaborate when we do find risk factors.
Great. All right, next question here. This attendee mentioned, thank you for this nice talk. Humanity is maybe overrepresented on Earth, and I'm surprised that we carry so many mutations that could kill us. But could some of them represent a selective advantage over others?
Yes, and you probably are aware of the classic example of that, which is the sickle cell trait offers some selective advantage in malaria regions so that individuals can get sick with malaria, sick with sickle cell, but carriers will also be more resistant to malaria. And there are a number of examples like that. Other than that, it seems like there are a lot of mutations that occur spontaneously and are carried through populations, usually as a result of population bottlenecks, like the Ashkenazi Jewish mutations that seem by chance to have propagated within particular groups. I'm going to go into the next one.
Yep, we can go right to the next one. Sorry about that. I froze for a second there. We did also get a few questions in our live chat box as well. So to go over one in here, this question asks, as there is an additional cost, wouldn't an early intervention of cases such as cerebral palsy be cheaper in the long run as treatment, causes better physical outcomes, which requires less long-term equipment and therapies?
Yes, thank you. Perhaps the questioner is referring to cerebral palsy and the possibility of in utero treatment as opposed to newborn treatment, when presumably cerebral palsy would have already been established through some sort of damage to the brain. Absolutely. There will be categories of disease that lend themselves to in utero treatment. In fact, you could go back before conception and you could say, well, if parents were willing to screen themselves for recessive carriers fairly comprehensively, and then they were willing to use reproductive technologies to avoid having a child with a biallelic recessive disease, that would eliminate many of the conditions that we're talking about. So as the questioner implies, you can intervene pre-conception, during pregnancy, or at any point after birth. We just think that, and there's validity to all of those. We think that newborn sequencing happens to be one place that resonates sort of sociologically, psychologically. You have a new baby, you want that baby to have the best chance of life. You're already, if you're aware of it, you're already getting screened for conventional newborn screening. So we hope that this becomes a place where both parents and the medical system will find this more accepting.
Great. Next question here. Was there any negative feedback from parents? And if so, what did that look like?
Thanks. And there's also a similar question in the group chat where they ask, are you doing follow-up studies with parents of babies to find if there's any difference in distress over time? We did not formally administer those validated scales of distress, but all our other research, for example, for 15 years, we've been tracking distress in other types of genetic disclosure. And in every single one of them, if there was even a modicum of distress, it diminished over time. And certainly anecdotally, these parents have not a single one has articulated increased distress over time. So I feel pretty confident in saying that there was, in this fairly limited sample size, of course, not only no observable distress in the short term, but none in the long term as well.
Great. We have time for a few more questions here, so we'll try to get a few more answered. This next question asks, as the title of your presentation states towards universal sequencing, how do we make sequencing for the target genes identified by your research universal?
Yeah, and it's a really interesting question because perhaps listeners can interpret universal two ways. There are 20,000 genes, of which about 7,000 are well-established disease association genes. You might think, oh, universal genome sequencing means actually interpreting all 7,000 of those genes. And that's not the sense in which we're using it. I'm using the term universal to mean, could we envision a day when every child, every parent has the option to have their child inexpensively or for free tested for genomic conditions along with the conventional metabolic newborn screening that they get today? Could we imagine a situation, a scenario, an infrastructure, a cost structure where this is possible? And I believe that we can. I believe that it'll look very different than our current newborn screening system. I don't think 53 state labs should be the way that this rolls out, but I do think we can envision that, and I do think it will save lives. And so that's the aspiration, the goal, and the dream that I'm using to title the talk.
Great. Thank you, Dr. Green. It looks like we have time for one more question here, so we'll wrap up after this one. How feasible is WGS for newborns now based on cost for countries in the global south?
Well, thank you for that question. I think it speaks to a lot of issues, including the larger issue of health disparities. As so often happens in technology, I think this will roll out first to information-seeking families who can afford it and who want this information. And they will both bear the brunt of the early expense, but also bear the brunt of any early risks, any early threats to privacy and discrimination. And then hopefully, as I'm sure the audience knows, the price of both sequencing and interpretation is being driven down at a tremendous rate. And we can actually envision a day when the entire process of newborn sequencing and a panel of, let's say, 1,000 genes being interpreted can be done accurately and at scale for less than $100, perhaps less than $50, perhaps less than $25. So I think we are imagining a day not too far away when these costs dramatically come down. And although there'll be cultural differences around the world in a willingness to do this, where the technology will quickly spread. When I think about this, I think about cell phones. I think about how the earliest cell phones were very expensive and only a few wealthy people could afford them. But very quickly, these came down in price to where they are today. Certainly not everyone can afford them today, but masses can afford them. And even countries around the world that are less resourced, less well-resourced, have often skipped over landlines and have a huge number of citizens carrying cell phones. So if you imagine the same thing happening with biotechnology, and sequencing and screening being a subsection of that biotechnology, you can imagine this spreading around the world, including traditionally less well-resourced countries at a rapid rate, certainly within the next 5 to 10 years. So I do think there's going to be a period of unrest. It's kind of a wild west in the in this arena, but I think this is going to be a dream that we can look forward to happening within the rest of my career.
Wonderful. Well, thank you, Dr. Green. Do you have any final comments for our audience?
Well, I do think that this is going to be a rough and tumble area. I think that it's like they say, this cliche, but still it's true. I think this is a paradigm shift. It's moving. It's forcing us to think, what do we want to screen for, and what do we want medicine to do for us? Do we only want medicine, or primarily want medicine to treat us when we're sick, or do we want medicine to give us information into the future that will allow us to individually follow the path that keeps each of us the most healthy? And there is an enormous number of forces, from the traditional educational track of doctors to the incentive structure to payers, to the built-in ecosystem of device manufacturers, hospitals, and their operating rooms and emergency rooms that are invested in the current system. So it's not new. You've heard this message before, but it is rather revolutionary to propose something that's truly going to shake this system up. And I encourage everyone to embrace this possibility, not be afraid of it. We're never going to have all the data we need in each of these rare diseases in order to begin the process of screening. We're going to have to begin the process of screening and improve that process as we go along. And I think the challenge is to find ways to do that responsibly, but recognize that it must be done sooner rather than later. And I hope I've convinced you through this talk to join forces with us on that.
Awesome. Well, thank you again, Dr. Green, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until May 18th, 2025. Labroots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.